Good day to everyone joining us, and welcome to today's Xtalks webinar. Today's talk is entitled RBQM 101, Getting Started with Risk-Based Quality Management. My name is Sonia Hunt, and it's my pleasure to be your Xtalks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank ARG who developed the content for this presentation. ARG is a contract research organization focused on oncology, immunology, neurology, providing comprehensive clinical program development services ranging from pre-launch consulting to commercialization, achieving over 50 market authorizations to date. Founded in 2004 with the vision that every project should be highly individualized, ARG has experienced consistent growth across the globe, expanding their reach to include drug and device strategic consulting, clinical trial management services, and clinical data and analytical solutions. ARG uses first-in-class technology platforms along with relationship-driven flexibility to optimize clinical studies because they believe everyone deserves to be well. Now it's my pleasure to introduce you to your speakers for today's webinar. And first, I'd like to introduce you to Erin Farris. She is the Vice President of Operations at Atlantic Research Group, responsible for overseeing project management, clinical operations, and medical monitoring services. Erin, along with her leadership team, provides the training and infrastructure necessary for individual contributors to be successful in performing the task at hand. Her team is focused on the efficient and streamlined approach to risk-based quality management using robust data analytics to help teams make informed decisions. Erin holds a BS in biology with a minor in chemistry from Virginia Tech. Next, I'd like to introduce you to Brad Whitlow. He is a project director at Atlantic Research Group, responsible for directing and leading global project activities across therapeutic areas, ranging from early development to larger scale projects or programs. Brad provides operational oversight across multiple clinical trials, ensuring assigned project managers and project teams are meeting ARG and study requirements. Brad has over 20 years of experience spanning research hospitals, biopharmaceuticals, and CRO companies. And finally, I'd like to introduce you to Lauren Garson. She joined Veeva in 2018 and is a senior director in the Vault Clinical Operations Strategy team. She is responsible for Vault eTMF, SSU, CTMS, and payments for enterprise-level customers, and is the lead strategist for the TMF transfer and risk-based study management features in the Clinical Operations Vault. She has a BA in Human Development and Family Studies from Cornell University. And now it's my pleasure to pass over the mic to Erin. So Erin, when you're ready, you may begin.
Thank you, Sonia. Hello, everyone, and welcome. We hope that by the end of this webinar, you will learn why you shouldn't put off RBQM, the best practices, challenges, and solutions, how to develop key risk indicators, or KRIs, aggregate data, and establish thresholds. And then also tips to get started today with the technology that your company has available. Next slide, please. So first, let's define risk-based quality management, or RBQM. RBQM applies principles of risk-based monitoring to the study in its entirety. It uses technology, analytics, and statistics to detect data quality issues in real time. This concept is not new. In August 2013, the FDA issued a guidance document to support a risk-based approach to monitoring. It provides the rationale and framework that researchers can put into practice. Furthermore, the ICH E6R2 states the sponsor should develop a systemic, prioritized, risk-based approach to monitoring clinical trials. This can be on-site monitoring, a combination of on-site and centralized monitoring, or where justified, just centralized monitoring. So Sonia, this comes to our first poll question, please.
Certainly, I am going to launch our first poll question, and the audience members should see it appear in front of their screens right now, and there it is. So please go ahead. This is done in real time, so your participation is strongly encouraged and very much appreciated. So please select from the below, and the question we have is, what is your level of experience with RBQM? Okay, and the options are none, novice, intermediate, or expert, or novice, sorry, intermediate, and expert. Okay, I'll give you another 10 seconds to complete this poll, as it looks like we have the majority of our audience have already cast their vote. So thank you very much. A few more seconds for those of you who have not, if you can do so now, and then I will close the polls and we'll get right back to Erin. Okay, that's it. Thank you so much, everyone. Let's take a look at the results. Okay, so here are the results. We have here 22% that said none, 33% that said novice, and then we have 38% that said intermediate, and 7% said expert. So thank you everyone for participating in our first poll question. Now back to Erin. And Erin, before you begin, what do you think about the results of that poll question?
I found it quite interesting. It looks like we have a variety of experience in our attendees today, which is great. Hopefully it will fuel some wonderful question and answer session at the end. So it takes me to the next presenter, who's going to talk a little bit more about adoption of RBQM. Lauren?
Thank you, Erin. So let's talk about some key challenges for adoption. Regulatory requirements around RBQM have been around for a long time, and they give a good idea of what you should do, but not how you should do it. With the emergence of new technologies for data collection, the continued impact of the COVID-19 pandemic, and the increasing number of partially or fully decentralized clinical trials, the importance of risk-based monitoring, or RBM, and the larger risk-based quality management, or RBQM framework in clinical trial management is increasing. Despite a growing body of evidence showing the benefits of RBM in clinical trial management, adoption has been slow and implementation incomplete. So let's talk a little about why it's a challenge for customers to start adopting RBQM processes in their organizations. So to help with some of these challenges, a lot of technology companies and consulting companies have come up with solutions to help meet the RBQM need, but disconnected point solutions can make things too difficult. So those who work in clinical operations like study managers and CRAs shouldn't have to have a statistical analytics background to get to the information they need to get their job done and understand where a risk occurred in a trial. There's so much data to analyze. How do you determine which are the critical data variables that could provide the most risk to your study? And a lot of companies have decided to use Excel to complete many of the RBQM tasks, but Excel, as we know, is not meant to do the job of running a clinical trial. Sonia, if you can go to the next slide. So RBM in clinical trials focuses on detecting, addressing, preventing, and mitigating risks that could compromise clinical trial processes, patient safety, or data integrity. This more efficient approach to trial monitoring is an integral part of the RBQM framework. Completing the risk assessment is the foundation for greater adoption of RBQM. So defining those critical data and processes and the associated risks early during trial planning promotes the adoption of other components, including risk mitigation through centralized monitoring. So later on in the presentation, you're going to hear Erin and Brad, and they're going to be discussing how ARG completes and communicates their centralized monitoring process, which enables the reduction of costly on-site monitoring by allowing you to focus on those data points that matter. This more holistic view of the clinical trial data allows on-site monitoring efforts to be targeted based on the output of the centralized monitoring process. So that will increase your monitoring efficiency. It pulls together data collected from different sources to provide a holistic view, aggregating the data to identify compliance and deviations and trends critical to subject safety and data integrity. You go on to the next slide, Sonia. So if RBM and RBQM is the way that regulatory agencies tell us we should be running our trials, Why do we need separate tools to do those specific processes? And as I mentioned before, why use Excel, which is a basic tracking tool and not built specifically for the RBQM use case? Why not embed the RBQM processes within the same systems your teams currently use to run your trials, like a CTMS? This is going to make things much easier for you. And since your teams are used to using CTMS for their other processes, adoption of these processes will increase. So on one side of the slide here, you're going to see a video of our Veeva tool within our CTMS. So at Veeva, we didn't build a specific tool for RBQM. We have it embedded, a built for purpose risk assessment within Veeva CTMS. So we're trying to make it as easy as possible for you to meet the regulatory requirements of RBQM. This allows your teams to enable the use of risk libraries and risk templates, so they don't have to reinvent the wheel each time they complete a study risk assessment. If a study is similar to another study, you can reuse the same template or pull down your oncology-related risks or specific country or site-related risks. And you can document and track any risk mitigations to completion using your Vault CTMS standard issue management capability. So it's all there to make you more efficient. And the process is also embedded with configurable review and approval workflows that are tracked, so you know where you are in your risk assessment process and who is doing what part of the assessment. And when your risk assessment is complete, you can automatically file your risk assessment document and all future versions of that risk assessment within the appropriate place in your TMF to be inspection ready at all times. And one more thing, because Vault is a relational database, another differentiator from Excel, you can track trends across your entire portfolio. So where have you used a risk before? How was it assessed? This is a great lessons learned perspective. Sonia, I think next we have another poll question.
That is right, and I'm going to launch our second poll question. Audience members, you should see it now, and there it is. So just like we did before, this is done in real time. I'll give you 45 seconds to complete this simple poll question. And the question is, what is your organization's current percentage level of RBQM adoption? That easy. So please go ahead and select one of the below: 0, 25%, 50%, 75%, or 100%. Okay, and talking about percentages, it looks like we got about 60% of our audience members have voted, so I'll leave another couple of seconds for those of you who have not. If you can do so now, it'd be greatly appreciated. Okay, that looks like it's the majority of everyone, so thank you everyone for participating. Now let's just take a look at the results. Okay, so here are the results. We have 17% are saying 0, 25% of our audience members are saying 25%, 36% of our audience is saying 50, and then we have 6% that are saying 75, and 17% of our audience is saying 100. So thank you very much, everyone, for participating in that last poll question. Now I'm going to return it over to Lauren. But before Lauren begins, Lauren, what do you think about the results of that poll question?
I think that's really interesting. Just like Erin's answer earlier, there's a varied response from our attendees, and we hope we're going to get that 0% up to 25 or 50 or even up to 100% after you guys are listening to the presentation. So I'm going to pass it off to Brad, who can talk a little bit more about the risk assessment.
Thank you, Lauren. Next slide, please. Yeah, so I'm going to start out and give just a high-level overview of risk management in general. And based on those first two poll questions, it seems that we do have some attendees who have ranked themselves as expert and 100% knowledge within your company. But we do have quite a few folks that seem just to be starting out in this process or hoping to gain some more knowledge. And that's what we're going to talk about over the next couple slides and what you can implement right now in your organization. So in regards to risk management, you'll see in this simple flow diagram, we have the process, and the process starts with simply identification of those risks. Next, we assess risks. After identification and assessment, we then come up with mitigation strategies in order to put in place to be proactive in case a risk does happen or occur, we have a mitigation already in place. We review all of these identified risks and assessments and mitigations on a predetermined frequency. And then last but not least, we have to report what we have found and report those reviews as we track these. It's kind of the old adage in clinical trials, right? That if it's not documented, it didn't happen. So this is where the risk categorization tool comes into play. Next slide, Sonia. So again, this is a simple example of a risk assessment categorization tool, commonly referred to as RACT. And within this tool, you will see that we have identified the risk from the category in the fourth column there. And there's a pull-down menu that lists the different categories, examples of different categories that you can choose from in which particular risks would fall into as you move forward. And then you define or describe the risk. And so it's simply, the first step would simply just be to get together with your internal team. It's almost like a brainstorming. It is a brainstorming session where you review a protocol and go through all the potential risks that you think may occur or the risks that are presented by your particular protocol. We then would strongly suggest a protocol risk assessment meeting with your sponsor, just to get on the same page and feel out what the sponsor feels are priorities in the risks that they want to track through their risk assessment categorization tool. And then next, what we're going to talk about is assessment and mitigation of those identified risks. So next slide, please. So with the assessment and the mitigation, what we do and what you should do with each risk that you identify, you next walk through this process. You look at the probability of a risk occurring during any time in your study and what impact that could have on your study. And then in this example, the severity of that risk is a simple formula of probability plus impact multiplied by 2. So again, on the screen, what you're seeing is for this, any potential risk, we decided this particular risk was a medium probability, had a medium probability of occurring. And what impact, if it did occur, what impact would it have on the clinical study? And again, we selected 3. And based on the formula, that gives us a high risk score. Then in the next column, next to your severity, we then show documentation of what mitigation strategies can be implemented either to deal with the risk at hand or lessen the effect of the impact as it occurs. You will do this throughout the entire trial. As the process diagram showed, it's just not a one and done process. You are continually assessing risks throughout your study, because as you go along in the trial, some risks may disappear and are not, after you get done with startup, maybe those startup risks, those are done. You're into the maintenance of your study. Or what you can find as well is maybe a risk that you assessed at the beginning as high is no longer high. It may default, it may drop down to a medium risk level in which you would maybe implement a different sort of mitigation strategy. Next slide, please, Sonia. I believe we have a poll question number 3 coming up, please.
Certainly, Brad. I'm going to launch it right now. And our audience members, they already know what to do. I'll give you 45 seconds again. Just please go ahead and select one of the below. And the question that we have for you is, what is your organization's main barrier to adoption? Regulatory guidance issues is an option. System and budget requirements terminology and process uncertainty, key risk indicator, development and management, or all of the above. Okay, I'm going to leave a couple more seconds. We still have a few people there who have not cast their votes, so I'm just going to leave a few more seconds for them to go ahead and do so. Okay, that's looking like it's the majority. A few more seconds, and then I'm going to close the polls, everyone. Okay, perfect. Thank you, everyone. I'm now going to close the polls, and let's take a look at the results. Okay, so the results are here. We have 8% of our audience are saying regulatory guidance issues, 31% are saying system and budget requirements, 11% are saying terminology and process uncertainty, and we have 6% that are saying key risk indicator, development and management, and then we got 44% of our audience that are saying all of the above. So thank you everyone for participating in our final poll question. Brad, what do you think about the results of that poll question? Is that something?
Yeah, it's pretty indicative in what I would expect, especially the 31% in budget requirements. And that is exactly what we see and why we're talking this morning, and what Erin is going to touch on next, on using the tools, whether that's an Excel spreadsheet or something similar to you, that can get you up and running today instead of putting it off. Just kind of putting your toe in the water and getting some experience on RBQM. So with that, Erin's going to talk a little bit more about centralized statistical monitoring. Erin?
Thank you, Brad. So Centralized Statistical Monitoring, or CSM, as I will be referring to, is ARG's interpretation and implementation of RBQM through the use of technology, analytics, and statistics to detect data quality issues in real time. So first, we need to take into consideration all source types. So you're looking at protocol specifications, as Brad had mentioned earlier. You're looking at the systems that you will be using, such as the clinical database or EDC, the clinical trial management system, safety system, ETMF, or even any vendors and the systems and procedures and processes that they will be using to provide a service for the study. You also need to look at types of risk. So we will be looking at the quality of the data that's coming in, quality at a site level, subject compliance, study objectives, and just the overall functionality of implementing the procedures and processes that had been put in place. Next is frequency. So the frequency in which CSM is needed really depends on the study needs. For example, monthly may be the best based on the quantity and frequency of data that you are receiving. Think of a study that has a high enrollment rate, for example. But you should also consider the timing as it relates to an upcoming data deliverable, such as database lock, or as we prepare for tables, listings, and figures for your clinical study report, or a safety monitoring board meeting. So all of these are things that could certainly impact when you decide to put this into place and into motion. And then lastly is the level of involvement. So this is a cross-functional activity, and we involve all of our key departments and stakeholders, everyone from biostats and data management to clinical operations, project management, drug safety, and medical monitoring. And the intent here is that everybody's going to be looking at this data with the different perspectives. So we want to make sure that it is very well thought through. And the next few slides will demonstrate just how we've put CSM into practice here at ARG. Next slide, please. CSM requires planning, and the first step is identifying your key risk indicators. I've highlighted some standard KRIs, and more can be added depending on the study. I noticed in the poll question, some people didn't quite know the terminology or how to get started. When you start researching things on the internet, you will be surprised at how many pieces of information you will find that give you a framework to start with on KRIs. I've just included a couple of these because they're pretty standard and apply to all projects across the board. You can look at under or over recruitment, rate of protocol deviations. You can even go into the types, so anything that involves informed consent or inclusion-exclusion criteria, treatment or out-of-window visits, for example, the rate of dropouts. Now you want to look at the percentage of data that you expect but you do not have, so missing data, rate of queries, over and under reporting of adverse events or serious adverse events. And then of course you always want to look at your endpoint data, whether you have primary or secondary. After all KRIs are identified, it's important to set thresholds that are study specific. Our project team determines what is acceptable versus unacceptable for each KRI. Let's take visit windows, for example. If a protocol specifies a visit every 21 days and they have a plus or minus 3-day window, then the threshold will account for this. And anything outside of that threshold will be flagged for attention. The third step in planning is deciding on the output. CSM outputs can be driven by a number of circumstances. As I mentioned before, you can look at enrollment rate or data-driven deliverables. The study team will need to decide on the frequency and expectation of how the data will be presented, reviewed, and discussed. Next slide, please. So after planning is complete, you are ready to implement. At ARG, our information technology and biostats team support the setup and implementation steps by making sure that data is extracted from the source systems, formulas and conditional formatting is programmatically applied based on the KRIs and thresholds that have been identified, and that the output is an accurate representation of these steps. The output can be as simple as an Excel file or as sophisticated as an RBQM statistical monitoring tool. Regardless of what you choose to use, it's important to note that continuous assessment and evaluation of these outputs and necessary may lead the team to modify KRIs, but also the thresholds based on the data trends and your study progress. Next slide, please. So this is an example of a CSM output as an Excel file. Each row represents the site-specific data, making it easier for us to compare site to site. The data within each row is a roll-up of that site's data, which is used to drive the KRI risk level. The columns are specific KRIs, and this is based on what the team has decided to track and manage, as well as an overall risk score. Risk levels are based on the KRI thresholds and are illustrated by red, yellow, and green filled cells. Red and yellow indicate that the acceptable threshold has been exceeded and action needs to be taken. Green signifies that data is within the acceptable threshold for that KRI. So the actionable risk levels direct you to the data that requires your immediate attention. Next slide, please. So I'm going to walk through an example of an actionable risk that was noted on the previous slide. Out of window visits. We want to evaluate the data making this KRI red, as shown in the first picture. So within the output, or the Excel file that we have the ability to drill down into the source data tab to look at which visit is out of window and by how many days. So this is illustrated in the second and third pictures on your screen. Upon reviewing this information, the clinical team is able to evaluate and document the action taken. For example, this out of window visit may lead to a protocol deviation that needs to be documented in the system. This decision is captured in this output and then carried over to the next output. So you have an audit trail, so to speak, of your review and decisions. And that is illustrated in the final picture called Evaluate. Having an all-in-one CSM output like this allows our project teams the ability to drill down into the source data, review actionable risks quickly and efficiently, and evaluate this at a holistic level. So in the next slide, Brad is going to share some of our success stories.
Thank you, Erin. By implementing some of these strategies that we've covered this morning, I have listed here just 3 study examples that we have found given feedback to us. And one, at the end of a study, there were really very few, or even in some examples, no questions or queries prior to database lock, because we have been managing and evaluating these risks all the way through the trial. So it made our lock very, very efficient. It also fostered trust between us, ARG as a CRO, and our sponsors. All of this information is being transparent and is shared with our sponsors as we go along. So when we got to a point at the end of the study, when we were generating statistical tables, listings, and figures, nobody felt blindsided. We were all on the same page. There was nothing, there was no hidden data or information that came up at the very end that could delay moving into the next steps. In early investigation, which is what all this is about. It's about being more proactive than reactive. So we identify these issues early on in the study. The sooner you identify them, the sooner you mitigate them and put a preventative action in place so they don't occur later on in the study or continue to occur. And so that too is generating fewer questions on the tables and listings and figures. And then finally, as I've already mentioned as well, a couple talking points were the fewer data-related questions. It just leads to a quick database lock and early deliverables of the statistical analysis. And everybody, with the transparency of the data availability, all team members, internal and external, are very aware of the data quality throughout the life of the study. Next slide, please. So this is what internally, some takeaways in what our staff has liked and how it creates some efficiencies internally. It definitely helps facilitate the ongoing communication from our internal team, from bios, data management, the clinical team. It enforces the messaging across our different functional areas. So we're all working on one thing, which is data quality. That's our goal, a nice, clean deliverable at the end. And that in turn decreases the chance of a timeline extension with nothing showing up as surprising at the end. Our staff, what we have done is seen a lot fewer emails. So if you have 200 to 300 queries and phone calls and emails to sites to get things cleaned up at the very end, you're really not wanting to rush around and do all this work as a study's wrapping up. That in turn makes it a lot less stressful. You know, the 2 biggest stress points in a clinical trial are startup and closeout. And when you're that close to closing out, you really don't want to be encountering all of these hidden areas of data quality, and that makes for a less stressful experience and just gives the team overall confidence in the data that has been collected. We also show the consistency throughout the study in the data trends. When we're looking at what Erin has described, part of that process is definitely showing trends in data, whether it's overall trends or site-specific trends, that we can focus more attention on those trends so they do not impact the data in a negative way. And it's been very beneficial to studies where consistency in data handling is an expectation. There's also this additional layer of review and oversight, especially in projects we've changed a protocol. If there's any major sort of revisions to a protocol, You go back and reassess your risks, and then you just continue with that same process as we go along. Next slide, Sonia. So with this, key takeaways, I'm going to pass back over to Lauren to summarize some additional key takeaways. Lauren?
Thank you, Brad. So we hope that you have learned a lot from this webinar, and we want you to realize that it can be easy to adopt RBQM into your clinical trials processes. So embedding RBQM tools directly with other tools you use, such as CTMS, can be a way to help with adoption across your organization. Combining your RBQM processes with your study management processes also will help with adoption. And also, it's good to make sure you outsource to the right experts that have done this many times before, such as ARG, to complete tasks that are needed to make you compliant with regulators and more efficient as well. So now I think we're going to open it up to questions. Right, Sonia?
That's right, and we're going to put on our webcams for that. So if I can get Erin, Brad, and Lauren to put on your webcam. There I am. Hi, everyone. Now, we are now going to begin the Q&A portion of the webinar. And for this segment, I would like to encourage our audience members to continue sending in their questions by using the questions window, which you'll find on the right-hand side of your screen. And we'll try to address your questions during the time that we have together. So Brad, Erin, and Lauren, are you ready for the first question? Okay, here it is. This audience member is asking, what kinds of studies should we start with? So who would like to start with that one?
I think I could take that. Go ahead, Brad.
No, go ahead, Lauren. You got it.
So I guess the easy answer to that is the next one that you have coming up, right? So there definitely are differentiators in terms of a study that you want to look at. Like if it's a very quick phase 1 study, you're not going to get as much of the benefit from RBQM. So doing it on a longer term study, you'll see more of the differences. But you really shouldn't hesitate. So if you have a study coming up, start early, maybe, contract with somebody like an ARG that has done this before so that you can learn on this first study, but that would be my answer. Brad, do you want to add anything else?
Yeah, that was exactly my sentiment as well. The next study you're doing, you should start. This need is to get in there and work this process. So yeah, the sooner the better.
Okay, thank you very much, Brad and Lauren. Let's go on to our next question. This audience member is asking, what kinds of risk libraries do you find useful? Who would like to start with that one?
Yeah, I can take that one, Sonia. I can start off. So risk libraries or risk categories. At ARG, I'm going to talk about risk categories, which is somewhat synonymous, but our risk categories, we build risk catalogs per therapeutic area. So we have a risk catalog for primary immunodeficiency disease. We have a risk catalog for oncology studies. We have one for neurological studies. So you have some basic risks that we can assign, build, and then build some mitigations within those different therapeutic areas. And then as a protocol comes in, then we can get more specific. But that's how we handle the different catalogs. We know that ARG being focused in rare disease and oncology, neurological diseases as well, we have built those out per our therapeutic experience.
Okay, thank you very much, Brad. Erin or Lauren?
Yeah, I will elaborate a little bit further on that. You can also apply lessons learned maybe in how a study is designed, or certain vendors that you're using, or processes to be able to build libraries specific to that. Obviously, if you have a blinded study, your risk libraries would look different than an open label, for example. So you can get a little creative, and then I would encourage lessons learned to help you reassess those libraries and continue to develop them.
Okay, thank you very much, Erin and Brad. Okay, this one is a little lengthy. This question is from one of the audience members who is asking, since all stakeholders will be reviewing the same set of data outputs, How do you ensure there is no gap in terms of the data reviewed from, for example, stats, code, medical monitoring, safety, etc.? Who would like to take a shot at that one?
I can. I
Thank
feel
you.
like that's right in my wheelhouse.
Okay.
So the example that I provided in the slides, that's Excel, we work within a Google platform so that we can collaborate. So the team gets together and reviews the same output together, makes comments, adds whatever actions within that document and assigns it to team members. And so it's very much a collaborative discussion. I would also encourage a meeting, at least for the first couple of times, among the team to review it together. And then you can get a feel for how each department is looking at that data. Because the way biostats is looking at the data is different from medical monitor, which is different from a clinical perspective. So that's how we've done it. It's been very successful.
Okay. Thank you very much, Erin. Anyone else like to add to that? Nope. Okay, let's go on to the next question. This audience member is asking, how do you anticipate ICH-R3 impacting the industry's adoption rate of RBQM? And do you feel many companies are perhaps waiting for ICH-R3? Who would like to start with that one?
I can take that one.
Okay, thank you, Lauren.
So I think it's my understanding that R3 is going to be focusing on a response to the changing types of studies that we have out there. So real world evidence and digital trials and innovative trial design. So there really shouldn't be a big impact to what the standard studies that you are running right now in terms of adopting RBQM. So you should not wait. There's no reason to wait. The regulatory authorities are really expecting trials to be run using these processes, but then not just from a regulatory requirements perspective, but also for you, it really will increase the quality of your study because you're focusing on those high risk areas. So there really is no need to do 100% SDV. There really is no need to treat every data point the same way. We've seen from experience that the way that you adopt these processes will make your studies have better quality. And then you will also, if you are inspected, that you will meet the regulatory requirements. So there really is no reason to wait. Erin or Brad, any other comments there?
Nothing additionally from me there.
Okay, you did it well there, Lauren. Thank you. All right, this looks like it's our final question. This audience member is asking, what roles should be involved in adopting and implementing RBQM? Who'll start with that one? Lauren, Erin, or Brad?
Well, I think I can address that, but I think Erin has definitely already hit on this, that the roles, it's very cross-functional, from your data management, BIOS, your clinical operations team, safety, QA, and your IT group. Everybody has a stake in this and a role to play. Including those sponsor team members coming together with our team members, reviewing all of those risks and all of that data. So it's definitely a team effort. It's just not for one specific, it's just not for the clinical operations folks to deal with. Everybody needs to be involved because, as Erin mentioned earlier, everybody has a different perspective on the trial and the data that trial is generating.
Okay, Erin or Lauren?
I would also add, yeah, I'll just add, if you have to pick one individual that might need to get it started at your organization, that would likely either be somebody in project management or clinical operations, or potentially even quality assurance would be my guess. But those have been the main functional areas that have been the drivers in helping ARG adopt this.
Anyone who likes to add? Lauren?
No, that's good.
Oh, okay. Okay then, thank you. All right, so that looks like, it looks like we have one more question coming in here. Let me just find it here. Okay, here we go. This question is, how do you maximize the use of Veeva for RBQM?
I think I'll take that one. So I think that there's really two main areas where you can get the most value of using RBQM within Vault CTMS. And one of them is that the processes are already embedded within your CTMS. So using the risk assessment, tracking your risks, using the libraries, a lot of the reuse there, and having everything tracked within a system so you can report on it afterwards is really a benefit, and you'll get the most value out of it. And then if you are also a Vault TMF customer, you can, once you complete your risk assessment within CTMS, it will generate a document and file it exactly within the correct place that it needs to be within the TMF. And then you can also do ongoing reviews of that risk assessment, and those will get filed, new versions of the risk assessment. So that's what I would see in terms of the biggest benefits of using and maximizing the use of Vault for RBQM.
Okay, well, thank you. Erin, Brad, Lauren, is there anything else you'd like to leave with the audience, or are we good to close out the Q&A now?
No, thank you for your thank you for your time and attention and your attendance.
Okay, perfect.
Thank you, ARG, for inviting Veeva to join with you guys.
Okay, thank you very much, everyone. And thank you very much to our audience members for sending in those questions. That ends the Q&A portion of the webinar. If we couldn't attend to your questions, the team at ARG will follow up with you after this presentation. And if you have any further questions, please direct them to Mike. And there's his email address right there. He'll be more than happy to assist you. Thank you, everyone, for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated, as it will help us to improve on our further webinars. Now, in a few seconds, I'm going to send you a link in your chat box. You'll be able to review the recording of this event at that link, and also share this link with your colleagues once they register for the recording as well. So I encourage you to do that. Now, please join us in thanking our speakers once again, Erin, Brad, and Lauren, for that very insightful presentation and for answering all your questions. So thank you again, Lauren, Brad, and Erin. Okay, we hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at Xtalks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone. Thank you. Bye.
Bye.